In the last few years, the center has changed in many ways. In addition to getting new systems, we've also got some new people in the facility who are highly skilled at microscopy. And one of the things we're trying to really emphasize in the facility these days is helping people conduct studies. So actually either providing complete support where people in our facility do the microscopy for investigators, or we help them design studies, or we even help them design complete new projects for supporting imaging studies. And that's a new focus for the facility. And so we're trying to encourage more people to come to the facility to utilize the services that we offer as opposed to just simply coming and using the equipment. This is the latest addition to our facility. This is a Leica SP8 confocal multiphoton microscope. And the thing that's exciting about it is that it supports incredibly high speed. It's based on a resonant scanner. So unlike a lot of other two-photon confocal microscope systems, it can collect images at 30, 40, 400 frames per second, very, very rapidly. The ability to collect images at that speed is fine. The problem generally is that you have very, very poor signal to noise at the high frame rates that these things support. So a critical part of the development of the system was the development of these new high D detectors that are very, very high sensitivity and very, very low noise. So it's really the integrated system that's capable of really taking advantage of the speed this thing offers. What this is for is primarily we're thinking of it for intravital microscopy, for dynamic events in intravital microscopy. So for example, looking at microvascular flow. The investigator that was most interested in this system is a pulmonary biologist who actually studies the lung in living animals. He can actually collect images of the moving lung in vivo. And so this system is ideal for that. And so we're very, very happy to have it and very excited about introducing it to our users. The reason that it's challenging to collect images in a living animal is that you have these problems of motion, obviously. To the degree an animal is moving because it's breathing and its heart is beating, all the things that are important in an animal, that makes it very difficult to collect images that have a resolution below a micron. If any motion in that kind of scale even will compromise the image quality. So the ability to collect images at 30 or 400 frames per second gets you around a lot of those problems induced by motion. Not only that, but the thing is, things like motion of the blood flow, for example, happens so fast. Another advantage of this is an investigator has used this to look at signaling in the heart, which is happening on at the millisecond time scale. He can capture transients of calcium waves in the heart of a living animal. This is a spinning disc confocal microscope. And the beauty of this system is that it's perfect for studies of living cells. The problem with fluorescence microscopy is that when you label a protein or a molecule in a cell with a fluorophore, so you can visualize it by microscopy, every time you look at it, every time you illuminate it, you destroy a little bit of it. And every time you illuminate it, you harm the cells a little bit. So it's a little bit like gardening if you're going to keep on digging up the seed every time to see how things are going. The more you look at it, the more you perturb the event. This is a microscope that is very, very gentle on cells. So if you want to do studies of living cells, you want to look how living cells are behaving in real time, this is the system to do that. The way that it works involves, there's something on the order of 1,200 points of light being illuminated on the sample at a time that are simultaneously imaged onto a camera, giving you a very thin focal plane image of the cells. And because there's a very low density of photons, a very, very low dose of photons being used to image it, you can take images for hours at a time without harming the fluorophores or harming the cells. So for dynamic studies of living cells without perturbing the system, this is an ideal system. So on the stage of the microscope is a stage incubator where we have cells growing in culture, and they're maintained at 37 degrees, the temperature they want to be at, in a humidified environment with CO2, physiological conditions. So we maintain the cells there for hours, days, or even weeks. And that gives us the ability to watch the chronology of events happening either very rapidly, because we can collect images at 30 frames per second, or over the course of days and weeks. A lot of biological events happen over those periods of time. So the last piece of the puzzle is that there's a system that Nikon developed called the perfect focus system that maintains the focus on a given plane at a time. So what that means is that whereas normally, just because of slight amounts of drift in the gears of a microscope, things move out of focus over the course of hours, if not days and weeks. This maintains the focus so you can get stability. And then finally, it's an automated microscope, which means that we're able to visit multiple fields over and over again. So if you wanted to, for example, look at the effect of 4 different drugs, Well, what you could do is you can set up a dish that has 24 wells, and you could look at drug A, drug B, drug C, and have 6 replicates of each of them, and the microscope automatically will go to each well and collect images. And so over the course of hours or a weekend, you will have a full study with replicates and statistically valid study having been conducted.